The second test that we're going to do is the Bradford assay. There are multiple ways to evaluate the concentration of protein, and this is one of them. That is very simple. If you want to know more about the mechanism of the Bradford assay, you can read in your lab manual about this chemical, this dye. But the main idea that I want to remind you today is that if there is low concentration, they will be brown, and if there is a high concentration, they will be blue. I have to remind you that you need to be careful with the Bradford reagent. Similar to the Benedict reagent, you will have to have goggles. This is an actual erosive reagent, so you cannot dump it in the sink, and there will be a bottle that you can dump your Bradford reagent when you are done. And we are going to do something called serial dilutions. And I posted a video, but the main idea is you will have the same concentration of a liquid, and then you will have to add another one. And but you start mixing that one and taking a little bit of that one, and you add less, and then in the next tube you add less. Then you mix that tube and you move from 3 to 4 and you add less. And every time you keep adding and adding less, and watch the video. And the idea that here you are going to have a gradient of concentration. Here you are going to have a lot of the BSA. While here you're going to have very little. So the color of your test tube will definitely have to be different. And here, this is an important question that make sure that you read this carefully. Here we give you the values in milliliters because we want that you do the conversion. So which make sure that you study those conversions and you try to see which type of micropipette do you use. This one that is very tiny, this one that is an intermediate, or this one that is very big. So you have to do this conversion, yeah. And I didn't go into detail, but when you read the lab manual, you can understand much clearer. What I wanted to know is that you're making a gradient and in the test tubes, and they will have different coloration. And this technique is a serial dilution, and you have to watch the video to understand this technique that is very important not only in chemistry but in microbiology. And the next step that we're going to do is with the coloration of the ink, we can detect the degree of protein, but we have a technique that is called spectrophotometry. Measure the amount of light in chemical concentration, and this allows us to study the amount of concentration. In your lab manual, you can read a lot about this. I'm not going to get into detail in this video. Something that is also important is that you will use this equipment that is the spectrophotometer. It's exactly this one, and I'm just going to also post a video so you know how to use it. Finally, we are going to use this spectrophotometer as I said before. I already, as I said, I posted a video about that, about how to zero the machine using your blank. Just very quick. In the video they explain how to use this cuvette. What you're going to do is take your blank, the test tube that don't have BSA, and you deposit it here. And then as they show in the video, you can open the spectrophotometer and put it here. And you can, in the video, they also show you how to use it, then how to use this control panel. And here is the place where you use to zero your machine. And remember, as soon as you are done and you take this out, please remember to put this back in the test tube, just in case that you need to blank again your the machine. Sometimes you can you have no problem measuring all of them. However, the machine sometimes make a mistake, and you have to blank again your to use your blank to go to the zero, to find the zero again. So remember, as soon as you finish with your blank, put it back in your test tube, so you make sure that you don't waste your time and you have to repeat the experiment. Remember to put uh, the cuvette in the correct position. They explain in the video. This is very important. This is the indication how you have to put your. This triangle is show you where you are supposed to put your the orientation of the cuvette. Please be very careful where you hold your cuvette, because if your fingerprints are in the cuvette, you will create an artifact. Your data will be all biased. So very careful how you hold that, and it's very well explained in your lab manual. And again, when you are done with the experiment, you will find a place to put your solution and a place to put the waste. And every time we are only going to give you 2 cuvettes to measure the 7 cube. Every time that you finish, you use tap water. So you put back, as a reminder, you put back from the cuvette, you put back your thing, the liquid, and then you have this empty, but it will be dirty, so you have to use tap water twice, and one time clean it with ethanol. Do not use acetone. If not, this will be damaged, and we have to get rid of this cuvette. It's not a big deal, but we try to save them. All this data that you're going to collect in the machine, it will help. So you have the different concentration of the protein, and here you will start filling. The machine will give you an absorbance, and it will give you an absorbance. So for example, I don't know, this will be 1, this will be 3, this will be 4, like this. And with that, we are going to make a standard curve. So the idea is that there will be a point that when you compare the absorbance and the concentration, that they are proportional. The higher the concentration, the higher the absorbance, until there is a point that they will start bending. And in the next video, I will show you how to make your graph to upload your data into Moodle. If you have any questions, just let me know.